Hello, everyone. Thank you for joining this session. So my name is Adam Kalinowski, and I'm the president of Dystonia Europe, and I'm also a dystonia patient. I have been suffering from cervical dystonia for 17 years. I'm also a board member of the Polish Dystonia Association and founder of the Polish support group for people with dystonia. So Dystonia Europe was established 29 years ago, and it's an umbrella organization for national dystonia groups in Europe. And today we have 20 members from 23 member organizations from 20 countries across Europe. And dystonia is a neurological movement disorder characterized by muscle contractures that cause involuntary movements and abnormal posture, very often painful. There are many types of dystonia that can affect one part of the body, several parts, or can be generalized, so it's affecting whole body. And there are treatments available, but unfortunately there is no cure for dystonia. And finding the cure is actually something that dystonia patients dream about the most. Therefore, one of our mission is support the research. And because research can be the way of finding the cure, also improving existing treatments and finding new treatment options. Therefore, Dystonia Europe established the David Marsden Award in 2003 to stimulate research and give some boost for especially young scientists to work in the field of dystonia. And today is with us Dr. Susanne Knorr from University Hospital in Würzburg, Germany. Dr. Susanne is this year award winner. And we will talk now about patient and public involvement in the research and how it helps patients and their families and society in practice. So welcome, Dr. Knorr. Thank you for joining us, and please present us your research. Thank you.
Okay, thank you for your introduction, Adam. Today I want to talk about from bench to bedside, translate basic science into everyday clinical practice. I'm really pleased and honored to have received the David Marsden Award this year for my research, basic research topic. It's about the pathophysiology of early onset isolated dystonia in a dystonic rat model. But today I don't want to present all my data we were generating here from this paper because we don't have enough time. So I would like to use selected data from my research to show you why basic science is so important also for the patients. In my talk, I will focus on dystonia because this is my research field, and dystonia is a rare disease. It's defined as a movement disorder characterized by sustained or intermittent muscle contractions, causing often repetitive movements, postures, or both. On this example, I would like to show you how severe dystonia patients can be affected. And then a huge issue is that the pathophysiology, the reason behind dystonia, it's still unclear. And if we don't understand the disease, we cannot treat patients well. And exactly for this issue, to deepen our understanding in human diseases, animal models are excellent model organisms for this purpose. Animal models have the huge benefit that we can perform experiment on every biological level. If you focus on dystonia again, dystonia is a network disorder. This means not only one cell type, one protein, or one structure, or one connection is affected. Whole structures which are connected to each other seems to be affected. This also means that different cell types, different brain areas are involved. Therefore, dystonia research needs a whole organism to investigate the pathophysiology of dystonia. And with a good model organism, we can investigate on every biological level. And one level, for example, is the phenotype level. Here we can analyze the phenotype, so the behavior and also the symptoms of the animals. Here you see a transgenic dystonia rat, which harbors the same genetic mutation as DYT1 dystonia patients. But as you can see here, they move totally normal. They don't have dystonic symptoms. And if we would perform experiments with this model, we couldn't catch the whole disease because one important aspect is missing, the symptoms. In DYT1 dystonia, we have people who have the same genetic mutation but no symptoms. And these people, they don't need a treatment. But 30% of these people with the same genetic mutation have dystonic symptoms and are severely affected from dystonia. And this affected people need a treatment, and for this patient, we need a good animal model to perform experiments. In our lab, we could generate such a model. We used here the same genetic rodent model, but we triggered dystonic symptoms by environmental factor. Here we used the peripheral trauma, and what you see here, this animal shows dystonic symptoms, dystonia-like movements. Here the retraction and clenching of the hind limbs. The next level, for example, can be the large-scale network level. Here we wanted to modulate the brain network with deep brain stimulation. For dystonia patients, the brain stimulation is already described as a safe and effective treatment. What is DBS? DBS is a brain pacemaker which sends electrical impulses through an electrode into specific targets of the brain. And what we did, we also implanted electrodes in the brain of the animals and performed the brain stimulation. What we found was that DBS was improving dystonic symptoms and that DBS had a modulating effect on neural oscillation, which is a rhythmic electrical activity in the brain. What I wanted to show you on this level, that you can analyze, for example, neural oscillation in different brain areas, what you cannot do in patients. Or you can test different DBS treatment strategies, like different positions of the electrode in the brain, different stimulation parameters. And the best results can then be translated into the clinic. But there are much more levels you can analyze. For example, the microcircuitry level. You can analyze the dopamine transmission by microdialysis, which is an important neurotransmitter in the brain, which seems also to be involved in the pathophysiology of dystonia. On the level of the cellularity and the molecular biology, we can also perform experiments by analyzing cell populations, single cells, proteins, RNA, DNA, and much more. I hope I could show you as a basic scientist how important basic science is also for patients. Our aim is to understand the pathophysiology of the disease and to investigate different treatment strategies, to finally translate our findings into everyday clinical practice.
Thank you very much, Dr. Knorr, for your presentation. So now, sorry, I think we can move to questions. And I have some first question to you. Doing your research, do you have a feeling that patients or public or some other organizations supported you how in your work, and if so, what kind of support did you get?
Of course, for us as a for basic research, we need always a funding because it's also really expensive, and we also need the positions for it. It's always funded from the public often, and we also have a new research project where different countries from Europe are taking place at joining us, and also Dystonia Europe is also involved in this project, and it's always good to have a discussion between patients, between a medical doctor and basic scientists. So, yeah.
Okay, thank you. And so, is there any question from audience, maybe? Don't hesitate to ask. Okay, so then I continue with my questions. Do you have any suggestions for us, for patients, organizations? How can we improve supporting researchers in their work? What else can we do? What can be supportive for you to do your work in the research?
Of course, there's always the question how we can finance all the projects, and for us, this is one aspect, and I think also because dystonia, it's a rare disease, it's often no one knows dystonia and no one really supports it because, of course, Parkinson, multiple sclerosis are much more kind of famous and they get more support compared to dystonia research, for example. So it's important that we make this more public, and I think the organizations are important to make this more public. And of course, it's also important that maybe these organizations tell us what they really need, in which kind of research they're interested in, and this is, I think, also important for us.
Okay, so in your opinion, in what kind of way we can let researchers know what we are interested in, and what kind of research could be beneficial for patients? Should we contact like researchers directly, or?
I think, of course, we have the medical doctors. They have contact to the patients, and this is, of course, good. And in our hospital, for example, we are really well connected. We have different working groups. They are working with patients, doing research with the dystonia patients, then we have the basic scientists, and we are really well connected. I think this is one way to talk to medical doctors, what their needs, what they're interested in. And I think for Dystonia Europe, you're presenting always on the conferences, and I think this is also important, that we can sit together and discuss some stuff, maybe at conferences, at congresses, and so on.
Yeah, and I think events like we have now, it's a great opportunity to do that.